I'm going to, as I say, talk about the complexity of clinical trials in Prader-Willi syndrome. I think clinical trials have complexity which seems to increase with the requirements for high quality, appropriately increase with high quality standards for trials. But there are, of course, particular challenges when one's looking at trials in people, for example, with Prader-Willi syndrome. Now, the control of energy intake in the brain is becoming, if you like, increasingly complex in terms of how we think about this. For a long time, there's been a focus on what's been called homeostatic eating. That is the basic physiological control of energy intake and energy output. But we now increasingly recognize that there are two other components which are relating to higher brain functions. That is hedonic eating, eating for pleasure, and then also the concept of executive control or executive function, which is how the brain can decide whether to eat or not to eat, or even to, if you like, reinforce the choices of foods which may be driven not just for pleasure, but by the sense of need or by social circumstances. Now, I'm not going to spend a lot of time on these next three slides, but I've put them in really just to show you our understanding of the complexities of the control of energy intake are expanding rapidly. We know that following food intake, as it goes into the stomach and then to the intestine, and then after absorption into, for example, organs like the liver, the impact on the pancreas, and then eventually storing energy in adipose tissue, there are a whole sequence of hormonal and neural signals to the brain that inform the brain as to what we have eaten and what those consequences, the breakdown products of the food, actually are. Here you can see a schema of the brain and some of the areas which are involved. The hypothalamus in dark blue is the area where it's thought that this homeostatic or automatic control of energy intake resides, a bit like where, and quite close to, where things like blood pressure and respiration are controlled. But it is these higher cortical centers involved with reward and executive control, and these signals are then integrated to have an impact on food intake and energy expenditure. Now, this is a very brief slide. Clearly, there is a huge knowledge about the neural mechanisms which are thought to underlie hyperphagia in Prader-Willi syndrome. But I think I wanted to just emphasize that the results of the investigations do suggest that there are distinct neural mechanisms which are associated with the hyperphagia in Prader-Willi syndrome, and that these involve not just the hypothalamic homeostatic centers, but in particular, some of those centers involved with hedonics and control, as shown here in the second bullet point. Also, there is quite good evidence that one of the hormones secreted in response to food, and which is normally suppressed after eating food, ghrelin, the hunger hormone, that these levels are higher in people with Prader-Willi syndrome, and that the impact of ghrelin is to enhance some of the pathways involving dopamine, which are thought to be involved with reward and even, in some cases, addiction. Now, the hyperphagia questionnaire for clinical trials is used, and I'm sure you'll hear more about it in clinical trials, where hyperphagia is often the end point for evaluating a new pharmacotherapy. It's been derived from a larger questionnaire, and it does measure and capture differences in hyperphagia behaviors between people with Prader-Willi syndrome and those without. But of course, there are problems where the person with Prader-Willi syndrome may be living in a setting with highly structured food routines, where this hyperphagia score from the questionnaire may not fully reflect the extent of hyperphagia. And of course, Prader-Willi syndrome is much more than just hyperphagia. We know it's involved with a whole variety of behavioral changes, in terms of cognitive changes, issues such as anxiousness and rigidity. And of course, if we only measure hyperphagia, that's only measuring one small aspect of a potential for a new therapeutic agent. And so one of the challenges for clinical trials is, can we measure things beyond hyperphagia? Of course, we can measure weight or body mass index, but we can't do that if the weight is already in control. And it is, of course, a slow responder to any intervention. One may have to wait 3 months or even longer to see a change. Tempo outbursts, well, there are a number now of questionnaires or reporting systems to try and record these. The same is true for anxiousness and for obsessive compulsive behaviors. What we really are missing, perhaps, are good biological biomarkers that would indicate that the impact of a drug is having an impact on Prader-Willi syndrome behaviors and the changes, if you like, in appetite and hunger. I might say that we don't have good biological biomarkers in more common obesities not associated with genetic defects. Now, beyond that, of course, there is the issue of recruiting people with Prader-Willi syndrome and getting the support of both them and their carers. And a while back, the Prader-Willi Syndrome Clinical Trials Consortium did a survey, which some of you may already know about, in which they asked which were the most important factors to decide to participate in clinical trials and what factors would prevent you from participating in clinical trials. And the main conclusions were, or the most important factors were, that people felt comfortable that the study team were the most important factors as to whether to decide to participate in the trial. And in terms of preventing participation, quite appropriately, perhaps, concerns about long-term or rare side effects, but then practical issues such as traveling, the financial costs of participating would limit the ability to participate in future trials. So I will just sort of end by, if you like, coming back to this schema that Prader-Willi syndrome is more than hyperphagia. And I think in the future, we are going to be asking for information from clinical trials to see whether there is an impact not just on hyperphagia, but on behavior in general, and whether we can have and develop well-validated methods of measuring such outcomes.